Cluster analysis is a multivariate technique, and what that means is not just that you have a lot of variables or that you have a number of dependent variables and a number of independent variables. There's a more restrictive description of multivariate data, and what it really means is that you have a bunch of observations that are not formulated in a model, but that are assumed to be correlated. You have, just think of a bunch of X variables that have been gathered in some domain, but on different dimensions that are, we don't know what the patterns are. And you can structure it even more formally. You can say, well, for each row, each observation, let's say I'm collecting information on 3 different variables, such as for a person, waist, the measurement around the waist, measurement around the chest, and height. You would assume that you would find clusters of patterns among those variables that fit. Well, often the clusters or the patterns, the groups, are not as evident, and so that's what these clustering analysis techniques work, and they have something in common. What you're doing is, almost in all cases, you're computing some measure of the similarity between the observations. And when I say observation, think of a spreadsheet. I mean a record, a row, and you may have 7 different variables that have values recorded for each row. Okay, so what you need to do is, for each observation, somehow you need to be able, you have to have some algorithm to compute the center, the centroid. And if you can do that, if you can find some reasonable way to compute a centroid value for each observation, then you can start to compare the distances. That is, how far each, the patterns in these centroids, how far each observation, the centroid of each observation, is from the other ones. And you can look at it that way. So they all pretty much function on this notion of similarity or dissimilarity. I say distance. When they're apart, when they're far apart, then they're dissimilar. When they're closer together, they're similar. What does distance mean? Well, you can have Euclidean distance or straight line distance. You're not talking about 2 dimensions. If you have 5 variables, it's not a 2-dimensional space. So you have to be careful about how you conceptualize the separation of the centroids for each observation. When you can have 2, 3, 5, 10 variables on each observation. So there's Euclidean, which is the absolute distance regardless of the number of dimensions. There's Manhattan distance, which means you plot the distance between 2 points at right angles, as if you're walking the streets of Manhattan and you can only go blocks north, south, east, west. And there are various other ways to do it. Okay, so we're going to look at k-means. K-means is probably the most commonly used. What k-means looks at distance between groups or centroids, but with k-means you are required to specify the number of groups in advance. Okay, so you tell it, let's take a look here. We're going to use the iris data, and we're going to set the seed. So we do that, and we're going to make a copy of iris, which is always a good idea when we start modifying the data set. So we just assign iris to iris2. If you've forgotten, iris was about flowers, and there were 4 predictive variables, all numeric, and then we're predicting the classification into the species of iris. And there were 3 species, Virginica, Versicolor, and Setosa. So pretty reasonable to assume we've got 3 different groups here. And what we're going to do is we're going to perform a cluster analysis without species present, just on the basis of these values. And we would expect them to show up in 3 different groups. If the species are clearly separated in terms of these values, which we don't really know for sure, but so we're going to do it in 2 groups and 3 groups and see what the clustering looks like. And so we get rid of species by, in a data frame, if you assign a column to null, it gets rid of it. You could also delete it other ways too, but so now if we go back and we look at iris2 after we did that, species is gone. We don't have that variable anymore. And then you just run k-means on the data set and you specify the number of clusters. So we say, okay, what does it look like? What's the pattern look like with 3 clusters? And notice this is an assignment statement. When you enclose any statement in parentheses, it expresses the value as well as performs the assignment. If I did this same statement, but I did not put it inside parentheses, it would create this object and assign the results to this object, but we wouldn't see it unless we explicitly expressed it. When you any sort of assignment statement, though, it's a shortcut to see the results and perform the assignment. So we retain the fitted object, but we also see the generic results. And what do we see here? Well, it's showing us the group membership. So we have, when we say three clusters, it assigns 62 of the rows to one of them, 38 to the other, and 50 to the third. And it's giving us the means for each variable, which is not the same thing as the centroid. You're assigning the records to groups, not the individual variables. So this, however, clustering vector does show us by row the assignment to the 3 clusters. And the way the criterion or the fitting function for k-means is to minimize the sum of the squared error terms, the distances of each point from the centroid within each cluster. So it moves points to 3 different clusters such that the final membership, the final assignment of each point to the 3, which is fixed, clusters, minimizes the sum of the squared distances of each point from those centroids. And this is typical. Note this is not just cluster analysis. Reducing or minimizing the sum of the squared error terms is pretty much, applies to almost any sort of linear function or nonlinear function as well. You start calling it deviance, but you're still talking about the same thing, the difference between the predicted value and the actual value. Okay, so you minimize, specifically with k-means, you minimize the within cluster sums of squares by cluster. And that's the between sums of squares divided by the total sums of squares. So what does that mean? Well, cluster analysis is one of those techniques. It's really easier to take a look. What we're trying to do, though, of course, is a practical application. We're trying to correctly assign the records to the flowers with the flowers removed. So now we can take a look. We can tabulate the assignment, these assignments 1, 2, and 3 to the actual species. And we get this, which is very similar to a confusion matrix. What we see is just like we did with the random forests, Setosa is very easily and correctly predicted. Membership in the Setosa species And that's because the combination of the values for the variables that describe setosa are different. The centroid of those variable values for setosa is very different and distinct from versicolor and virginica. And versicolor doesn't do too badly, but virginica, it's not clear. By looking at this, you could argue that there are really only 2 clusters, not 3, because this one is blurred and really is split between the virginica and versicolor. So with cluster analysis, it's easier to plot it and take a look, and we can also plot the centroid of each cluster. So with points, so we here's the plot, and it's still 2 by 2, so I got to clear that out. Here's the plot, and then we add the points, which reflect the centroid. It looks like 3 groups, pretty much, except the black and the red are kind of mixed. And that's the versicolor and the other one, not setosa. The greens are the setosa, and they're clearly separate. Okay, but these 2, versicolor and virginica, are distinct a little bit, but not too much. But it's easier, it's helpful to plot the center. So we do that with points. Let me zoom this. These asterisks are the center of the centroids for each of the 3 clusters. And you can see easily that some of the black ones are closer to the center of this red, and some of the red ones are closer to the center of the black. But looking at it in 2 dimensions is misleading, because it's a 4-dimensional object, because you have 4 variables. So some of them come out, but nevertheless you can see that these are not clearly distinct, whereas the green ones all are. Green is setosa. So let's go with the 2. K-means in the hierarchical clustering is probably the most popular, and we're not going to go with the 2. Okay, so we'll leave it at that. We'll look at 2. We'll come back to that, and we'll look at 2 with another approach in just a second.